Welcome to another episode from the Veterinary Medicine Digest. Today, we're delving into the fascinating and sometimes challenging area of neurological disorders affecting the white matter of the brain and spinal cord in dogs. We'll be specifically discussing a group of conditions termed Leukoencephalomyelopathy, drawing on recent research that sheds light on distinct presentations in several breeds. White matter disorders are complex, involving the nerve fibers that carry signals throughout the central nervous system. Understanding these conditions is crucial for diagnosis, treatment, and ultimately, improving the health of affected dogs. We'll focus on 3 specific breeds where these conditions have been recently described or further characterized. The Leonberger, the Rottweiler, and the Border Terrier, examining the clinical signs, the findings from advanced imaging like magnetic resonance imaging, and the detailed changes seen upon microscopic examination of the affected tissues. This comparative look helps highlight both the shared features and the unique aspects of these breed-specific diseases. Before continuing, be sure to like, share, and subscribe as proceeds generated from this channel will be donated towards animals in need of veterinary care.
These disorders can present in different ways and often affect young to middle-aged dogs, leading to progressive problems with coordination and movement. For instance, a novel disorder has been identified in the Leonberger dog. Affected dogs typically present with initial signs around 2 to 2.5 years of age. The clinical signs often begin intermittently, but then worsen over a period of months. Owners might first notice spontaneous knuckling in the front limbs. As the disease progresses, dogs develop a more generalized lack of coordination, or ataxia. A characteristic finding is a hypermetric gait, particularly in the front limbs, where the dog appears to overreach. Proprioceptive deficits, meaning the dog has trouble knowing where its limbs are in space, are observed in all limbs. Some dogs show stiff or stilted steps, or drag their paws while walking. Despite these significant neurological issues, routine blood work and cerebrospinal fluid analysis are often found to be within normal limits. This lack of inflammatory changes on standard tests can help guide the diagnostic process away from infectious or inflammatory causes, although infiltrative diseases still need to be considered.
The clinical signs you described certainly point towards a problem in the pathways of the nervous system responsible for movement and coordination. This is where advanced imaging, such as magnetic resonance imaging, becomes incredibly valuable in the diagnostic workup for these cases in live animals. In the affected Leonberger dogs, the findings on magnetic resonance imaging were quite distinctive. The most obvious lesions appeared as areas with increased signal intensity on specific types of imaging sequences, like the T2 weighted images. These changes were primarily located within the dorsolateral funiculi of the spinal cord. A crucial observation was that these lesions were typically bilaterally symmetrical, meaning they affected both sides of the spinal cord equally at certain levels. They extended vertically, in one case from the 1st to the 4th cervical spinal cord segment, becoming less intense further down. In another case, the lesions were seen in the dorsolateral funiculi of the 2nd cervical segment, also symmetrical and with increased signal intensity on T2 weighted images. Importantly, these areas did not show any abnormal contrast enhancement after the administration of a contrast agent. The symmetrical nature of the lesions combined with the lack of contrast uptake strongly suggested a neurodegenerative disorder rather than an inflammatory one. This allowed clinicians to include conditions like leukodystrophy, neuroaxonal dystrophy, axonopathy, and leukoencephalomyelopathy in their list of differential diagnoses.
When we look at these cases after death, performing a necropsy, the macroscopic changes provide the initial clues about the disease's impact on the central nervous system. In the Leonberger dogs, the gross lesions were observed to be restricted mainly to the spinal cord and the brainstem. These appeared as opaque, whitish discolorations within the white matter, most strikingly in the lateral columns of the spinal cord. While often described as bilaterally symmetrical, it was noted that sometimes these discolorations could appear unilateral or have an asymmetrical pattern at certain levels. The lesions were most severe in the cervical region of the spinal cord, but could also be seen grossly throughout the thoracic spinal cord and extending forward into the pyramidal tracts of the brainstem. However, the true extent and nature of the disease are revealed through microscopic examination. Histologically, the hallmark finding in the affected areas was a marked loss of myelin. This was evident as a paler staining of the white matter in standard stains and a loss of the characteristic deep blue color with myelin-specific stains. Replacing the lost myelin was a prominent increase in glial cells, specifically a marked gliosis, with numerous reactive astrocytes that stained positively for glial fibrillary acidic protein. Although myelin was severely depleted, microscopic examination also showed that many axons were remarkably preserved, still running through the damaged regions. Wallerian degeneration, which indicates significant damage to the axon itself, was minimal. The presence of thin myelin sheets observed on more detailed sections suggested some attempts at remyelination by the nervous system's own repair mechanisms. This pattern, with significant myelin loss but preservation of axons, pointed strongly towards a primary problem with the myelin, or the cells that produce it, the oligodendrocytes.
The microscopic findings really solidified the suspicion of a demyelinating disorder. The observation of primarily myelin breakdown with relative axonal preservation is a key feature. While some swollen axons were noted, these were interpreted as secondary to the severe myelin loss, rather than the initial problem. Distinguishing between different types of myelin disorders is important. Dysmyelination, or leukodystrophy, involves a defect in myelin synthesis where the myelin is formed incorrectly and cannot be maintained. Myelinolytic diseases, on the other hand, involve the breakdown of myelin that was initially formed normally. Given the restricted distribution of the lesions and the later onset of clinical signs in these young adult Leonbergers, a myelinolytic disorder seemed more likely than a defect present from birth. However, the term leukoencephalomyelopathy was chosen as a broader term to describe this degenerative disorder, acknowledging the uncertainty about the exact underlying mechanism. The striking similarity in the history, clinical signs, and pathological lesions in two unrelated dogs of the same breed strongly suggests a genetic basis for this disorder. Although pedigree analysis didn't reveal common ancestors in these specific cases, and reported littermates were unaffected, the breed's specificity is a powerful indicator of a potential inherited condition. What's particularly interesting is the strong clinico-pathological similarity between this newly described disorder in Leonbergers and the previously recognized leukoencephalomyelopathy in Rottweiler dogs.
Yes, the parallels with the Rottweiler condition are quite compelling. Leukoencephalomyelopathy in Rottweilers has been recognized since the early 1980s. Similar to the Leonbergers, affected Rottweilers are typically young, with the age of onset varying between 1.5 and 3.5 years. The clinical signs are remarkably similar, including progressive ataxia affecting all four limbs, general proprioceptive deficits, and hypermetria, often described as a long-strided gait with stiffness and overreaching, particularly in the front limbs, which are frequently more severely affected than the hind limbs. Gender predilection has not been reported in Rottweilers. Progression of clinical signs occurs over months to up to a year, often leading to euthanasia due to difficulty walking. On magnetic resonance imaging in Rottweilers, bilaterally symmetrical hyperintensities on T2, weighted images are also observed. However, unlike the Leonbergers where the lesions were primarily restricted to the cervical spinal cord on imaging, the Rottweilers show these characteristic hyperintensities in both the brain and the spinal cord. In the brain, affected areas include the crus cerebri and pyramidal tracts. In the spinal cord, the lesions are located in the dorsal part of the lateral funiculi. These imaging findings, specifically the areas of increased signal intensity on T2, weighted images, corresponded directly to the areas of pathological change observed on gross and microscopic examination. The lack of abnormal contrast enhancement on magnetic resonance imaging is also a shared feature with the Leonberger cases.
Post-mortem findings in affected Rottweilers further underscore the similarities with the Leonberger disease. Grossly, there are often opaque, whitish, well-demarcated foci in the white matter of the cervical spinal cord and brainstem, particularly in the dorsal aspect of the lateral funiculi and pyramidal tracts, sometimes extending further down the spinal cord. Microscopically, the pathology in Rottweilers, like in Leonbergers, involves severe myelin loss and prominent astrogliosis. However, while myelin loss often exceeds axon loss, there can also be a decrease in the number of axons and some evidence of axonal degeneration, such as irregular profiles or mild swelling, which was interpreted as relatively mild. Attempts at remyelination are observed, though described as minimal or abortive, even involving Schwann cells near blood vessels in some areas of the spinal cord, which is unusual for the central nervous system and could suggest disruption of the normal barriers. The presence of preserved axons in the areas of severe demyelination in Rottweilers also supports the interpretation of a primary demyelination process. Although the exact cause is unknown, a genetic basis has also been suggested for the Rottweiler condition due to reported familial relationships among affected dogs, although the specific mode of inheritance remains undetermined. The restricted distribution of the lesions to certain functional systems in both Rottweilers and Leonbergers, despite being primarily demyelinating, is puzzling and has led to speculation that the primary defect might lie in the neurons or axons themselves, affecting their interaction with the myelin-producing cells. This hypothesis suggests that neurons might fail to provide the necessary signals to maintain the myelin sheets produced by the oligodendrocytes, leading to instability and breakdown. The Rottweiler cases, with evidence of both myelin and some axonal involvement and unusual remyelination patterns, provide an interesting model to study these neuron-myelin relationships.
Stepping away from the Leonbergers and Rottweilers for a moment, let's turn our attention to a distinct, newly described disorder affecting a different breed, Border Terrier puppies. This condition presents much earlier in life than the diseases in the other two breeds, affecting very young dogs shortly after they begin to walk, typically before 1 month of age, ranging from a few weeks to around 6 weeks old. The most prominent clinical sign is the presence of severe, generalized, coarse body tremors. These tremors have a low frequency and high amplitude and are often particularly noticeable in the hindquarters, causing a characteristic swinging side-to-side or rocking horse movement when the puppy attempts to walk. The tremors can be so severe that they significantly impair the puppy's ability to move normally. A key feature is that these tremors typically disappear completely when the puppy is sleeping or at rest. Cerebellar ataxia, or a lack of coordination originating from the cerebellum, was also noted. Neurological examination can be challenging due to the severity of the tremors, but typically reveals delayed proprioceptive positioning and slightly hypermetric hopping, suggesting involvement of sensory pathways and motor control. Unlike the adult-onset conditions, initial routine blood work in these puppies was considered normal for their age. Cerebrospinal fluid analysis in one case showed findings consistent with a mild to moderate non-suppurative inflammation, although the significance was unclear given potential age-related variations in puppy cerebrospinal fluid. Metabolic screening in that same puppy revealed findings suggestive of a possible error in metabolism, such as increased lactate and a positive test for mucopolysaccharides, although further specific tests like for certain enzyme deficiencies were not pursued.
The very early onset and the type of tremors clearly differentiate this Border Terrier condition from the later onset diseases in Leonbergers and Rottweilers. And the microscopic findings are also quite distinct. At necropsy, there were no obvious macroscopic abnormalities. The brain and spinal cord looked normal to the naked eye. However, histological evaluation revealed significant changes, but different in character from the primary demyelination seen in the other breeds. The key findings were a spongiform change affecting the white matter, essentially the presence of small vacuoles within the tissue, combined with a diminished myelin content. This combination was most severe in the cerebellum, brainstem, and spinal cord, with milder changes elsewhere in the brain. In the spinal cord, all white matter tracts were affected, but the severity was notably increased in the ventral funiculi. Similar to the Leonberger and Rottweiler cases, intact axons were present despite the abnormalities in the myelin and the spongiform changes. Importantly, there was a notable absence of microgliosis associated with these changes. The diminished myelin staining, or hypomyelination, combined with the spongy degeneration in the white matter, without evidence of significant inflammation, was interpreted as a spongy degeneration or spongiform leukoencephalomyelopathy. The presence of intact axons supported the idea that this was a primary myelin or myelin-producing cell defect rather than damage secondary to axonal loss. The combination of decreased myelin and spongy change, especially in the absence of microgliosis, suggests a complex issue potentially affecting both the synthesis and the stability of myelin. Comparing this to other described canine myelin disorders, while some hypomyelinating diseases present with similar early-onset tremors and involve decreased myelin, they typically lack the spongiform change seen in the Border Terriers. Similarly, other spongy degenerations described in other breeds, like the Samoyed or Australian cattle dog, might share the spongiform change but differ in the distribution of lesions or associated clinical signs. The specific combination of findings in the Border Terriers appears to be novel.
Bringing together these findings across the three breeds really highlights the spectrum of white matter diseases in dogs. While the Leonberger and Rottweiler conditions share significant similarities in their later age of onset, progressive clinical course characterized by ataxia and proprioceptive deficits, and the underlying pathology, which is primarily a demyelinating process with relative preservation of axons, they also have subtle differences in lesion distribution, particularly evident on magnetic resonance imaging. The Border Terrier condition stands apart with its very early onset, severe generalized tremors, and a distinct pathology involving spongiform change and diminished myelin content rather than overt demyelination and reactive gliosis. Despite these differences, a common thread running through the discussion of all three conditions is the strong suspicion of a hereditary basis. In the Border Terriers, the finding of a common ancestor several generations back in the pedigrees of the affected puppies, combined with the occurrence in multiple puppies from different litters with similar signs and pathology, strongly supports an inherited cause, likely with an autosomal recessive mode of inheritance. For the Rottweilers and Leonbergers, the breed specificity and occurrence in related individuals, though not always with clear inheritance patterns in the reported cases, also points towards a genetic predisposition.
The clinical presentation, especially the progressive nature and the type of neurological deficits, should raise suspicion for a white matter disorder in these breeds. Advanced imaging, particularly magnetic resonance imaging, is a critical tool for clinicians to help establish a presumptive diagnosis in live animals. The characteristic bilaterally symmetrical hyperintensities on T2-weighted images in specific areas of the brain and spinal cord, as seen in the Rottweilers, or primarily in the spinal cord as seen in the Leonbergers, are strong indicators of a likely degenerative process, especially when there is no contrast enhancement. The distribution seen on imaging often mirrors the areas of microscopic pathology. While imaging findings for the Border Terrier condition were not detailed in the presented information, the distinct clinical signs and early onset, along with the confirmed microscopic pathology, define this unique disorder. For all these disorders, a definitive diagnosis often relies on post-mortem neuropathological examination. The pathological findings, distinguishing between primary demyelination, axon loss, spongiform change, and hypomyelination, are essential for classifying these diseases. Understanding these differences is vital, not only for accurate diagnosis but also for informing prognosis and potentially guiding breeding decisions within affected lines to help reduce the incidence of these debilitating conditions. While the exact molecular causes remain unknown for many of these leukoencephalomyelopathies, ongoing research into their hereditary basis and the mechanisms of myelin formation and maintenance holds the key to future diagnostic tests and therapies.
Thank you for listening to the Veterinary Medicine Digest. A link for the reference paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.